Thank you very much. So this is an initial work we started doing in our lab in preparation for, in the future, doing the same work with near-field measurements. So I'll be talking a bit about a system here which we find very interesting to look at the near field, and also how in the far field we still manage to get some interesting information out of it. So in our lab we're trying to combine both abilities in material science, bringing interesting photoactive systems, combining them with interesting deposition systems, and then applying different optical measurements and hopefully some accurate models. So specifically, we combined here what's called J-aggregate nanorods, which I'll talk about more in the slides, with self-assembled, and we used hyperspectral measurements. So our base molecule here is the porphyrin molecule. For people who don't know it, it's a very interesting photoactive molecule, which has quite a high versatility. What's interesting about it is it acts as an electronic resonator. You have the pi bonds all around this main ring, and this gives you a lot of optical absorptions in the visible range, and both photoluminescence and even phosphorescence, both in all visible up to almost the IR. And what's very interesting about it is you can easily modify this system just by metallization. By basically putting a metallic ion in the center of the ring, you can completely change the optical properties. So for example, in our initial results, you can see that a non-metallized porphyrin has this spectrum. On the blue here, you see the absorption. On the right, you see the phosphorescence, the photoluminescence. And just by metallizing it with mangan, you see a very vast drop in photoluminescence, and you see a shift in the absorption spectrum. But metallization with zinc gives you also a very sharp photoabsorption peak and strong photoluminescence. So this molecule can be modified. What's more interesting about it is that if you take this molecule and you combine it, so you can, under certain conditions, this molecule tetrakrease 4-sulfonophenyl porphyrins, or TPPS4, I know it's a big mouthful, can form these structures, which looks like rolled up monolayers, but they're what we call them porphyrin J-aggregate nanorods. And these nanorods are not just like assemblies of this molecule, they're actually coherently coupled. So in the structure, you have coherently coupled structures of these molecules all together, and this can be seen just by looking at the spectrum, where you can see different vibrational peaks combined, both in the main peak of what's called the J band and the Q band peak, which usually has multiple vibrational branches, suddenly combines a single one. So we have here a result of a coherent coupling of many molecules, allowing them to decay all to the same level. Now, these structures are a prime material for near-field microscopy, since you have an interesting issue here. On the one hand, they're very thin, only 400 nanometer wide and only 19 nanometer high, but they're very, very long. So what we wanted to do with them is we wanted to couple them with plasmonic structures. But the problem we came with is that the coupling area was very small. We had this large array of plasmonic structures, but only a very small area, including only a few nanostructures were coupled. So using the standard technique of a laser spot, it was very difficult to get a meaningful signal out of this coupling region. So this is our first attempt to measure it. And here's our sample. We deposited these J-aggregates. What we did is we took a glass substrate with ITO coating. We used the e-beam lithography to create these nanodisk structures, which act as our plasmonic resonators. We chose these because we can control the resonance by changing the size or the distance between them. So we can either be on or off resonance with our excitonic molecule. And then we could cover them with a lipid peptide structure, which allows us to grow these nanorods directly onto our sample. And what we noticed, it was very interesting, we saw that they could either grow as this, it's very hard to see them in the SEM, but you can see either as a big cluster growing together from a single point. We saw single rods on our array. We also saw rods that cross into the array area. And we saw even rarely clusters on array, meaning a lot of nanorods grown together onto an array. So this gave us a large variety of different sample areas to look at. But as I said, the problem was to find the area of interaction. As you can see here, in the SEM, the rod only really sits on 1 or 2 of the structures in our array. The rest do not interact. So our first system we turn to, and hopefully in the future we'll do this again with near field microscopy, is hyperspectral microscopy, so I'll explain it a bit shortly. In a regular microscope, you have a light source, and you focus on a sample, and you image it onto a CCD. In a hyperspectral, we add an interferometer before the CCD, and instead of taking a regular picture, we take a picture of the interferogram of that sample. So you get a 3D interferogram, which you can then combine to get spectral data. So now you have a hyperspectral image of your sample using a far-field microscope, which is very interesting. Now, a method we used to sort all this data was case-mean spectral sorting. We basically wanted to locate the different areas where spectral reaction was the same, and to allow us to get a better understanding of our sample. So how the algorithm works, if you have here the image, false color image of our sample, and the center you see the array, and you see the nanorods crossing over the array, the algorithm takes along the main diagonal certain areas and decides these are clusters, areas of interest. And now I'm going to go and sort all of the pixels in the image. Remember, each pixel has full spectral data for that area because it's in hyperspectral measurement, and it sorts it to a cluster according to the distance between them in linear spectral space. Now, this sorting, of course, isn't correct. It doesn't tell us anything. But if we repeat this process several times, each time we take the new cluster, we sum all of the spectrum inside, and we find a new center, and then we resort the data. And if we repeat this enough times, and the algorithm actually converges, what you can see happens here. Slowly, the image becomes clearer, and eventually, it fits very nicely with our false color image on the other side. So now, not only that we categorized our different data in the hyperspectral image quickly, the different interaction zones, it also reveals interaction zones which are otherwise hard to detect. As example, this area here is, you can't see it really in the false color image, but you see it very well in this sorted data. You can see it has broken nanorods on it. So it has a partial interaction. It's not quite interacting as you can see here where you have the 2 peaks, but it's a shifted peak. So this is very interesting. So using this, we could quickly study all the areas where we had a single nanorod interacting with our sample. So instead of manually trying to locate areas, we could quickly scan and find all the areas where there was a single rod interaction. And here is the unmodified plasmonic resonance. You can see as we increase the size of the disk, we redshift the resonance peak. And after interacting, you see a smaller J-aggregate peak, which we categorize this as a weak interaction, weak coupling interaction. So the next stage is we wanted to see, okay, a single nanorod gives us a weak interaction. What about a lot of nanorods? So we looked at clusters. And before that, we wanted to basically construct some kind of model to explain what we were saying. So we entered the very simple quasi-static Rayleigh approximation model for a plasmonic resonator, which allows us to calculate its polarizability as dependent on the dielectric environment around it. And we added some corrections for long, large disks. And from that, it's very easy to find the scattering and absorption cross-sections for our plasmonic structures. So how did we account for the excitonic molecules? We added 2 corrections. The initial correction changes the substrate dielectric function by the amount of ITO coating on it, and we just calibrated that on a clean array. And the second correction, this F parameter basically is fitted according to the results after deposition the J-aggregate nanorods. And this lets us measure the amount of J-aggregate nanorods we see according to the change in spectrum. So we wanted to look at, as I said before, large interaction areas. So we looked for areas where there were clusters. And luckily, we had 2 very good clusters. One was on an array which was off resonance, meaning the resonance of the plasmonic structure was different than the excitonic molecule. And you can see it here at the AFM image, and here the false color image. And this cluster, once we ran the measurement and the algorithm, showed varying areas of interaction. So again, the colors here don't have any physical meaning. They're just telling you which cluster each spectral pixel represents. And what's interesting here, you can see in purple the clean structure. And in red, we added a reference peak just to remind you where the J-aggregate nanorod resonance is. And as you go towards the center of the structures, we saw a very interesting phenomena. What we expected was very similar to coupling. We expected both peaks to move away from each other as a result of Rabi splitting. But what we actually saw is that the J-aggregate peak was quite stationary. Even when it was much stronger, it remained in the same energy level, but a very strong redshifting of our resonance peak. And we inputted this data into our model and did fitting. Model showed the same results. So we said, okay, this is weird. Maybe it's because the off-resonance interaction is less strong. Let's go to a structure which is on resonance. So what did I do here? So we found one, luckily, that was also on resonance. And you can see here also a very nice cluster, even bigger than before. And we ran our algorithm. And again, if we scan from in outwards, very nicely, we again saw that the J-aggregate peak stays about the same area, but we have a massive redshift of our plasmonic resonance, up to 120 milli-electron volt from the original location. And again, when we inputted it, we saw that we have a linear increase in the F parameter. We can actually measure the change in thickness as we go towards the center of the cluster, and we correlated to this redshift we see in the plasmonic resonance. So what we hope to do in the future, and we're working on in the next couple of weeks, is now to correlate these results with near-field microscopy that should give us both the topographical and the spectral data in the same measurements. And hopefully we also have another question. We saw interesting results when J-aggregate crosses out of the array. We saw that the interaction, according to the hyperspectral, continues a bit beyond the area of the array. So seeing that also in near field with high resolution, we hope to see we can accurately measure how much of this coherence affects the interaction with the plasmonic particles. So I don't know the time, but I think I'm about finished. But the summary, the hyperspectral measurements helped us reveal hidden spectral variation in our samples, and created this system with a variable redshift plasmonic peak, and hopefully, and explained by a very relatively straightforward quasi-static model, and hopefully in the future, using the near-speak system, we can find out more about how these interactions happen. I want to thank both my advisors, Professor Shachar Richter and Professor Chaim Suchovsky, and our collaborators from Bar-Ilan University, Professor Zev Zevilensky and Dr. Itai Carmeli, and our funding, the Ministry of Science and Tel Aviv University Nano Center. Thank you very much. Thank you.